Okay, great. It looks like a fair number of people have joined, so we can get started. Hello, everyone. Thank you for joining the CADASIL Consortium this evening for our webinar on the genetics of CADASIL, presented by our very own University of Washington site principal investigator, Dr. Jayadev. Please input any questions you might have in the Q&A box at the end of the presentation. And without further ado, I will let Dr. Jayadev take the stage.
There we go. Thank you. Can you hear me? Great. Okay. So I'm going to share my screen. And here we go. Oops. Oh my goodness. Okay. Iman, does it look right?
I think we're seeing double screens right now. I think maybe use slideshow or swap displays.
Let's see here. Okay. Let's see here. I'll just do what I did before. How about that? And then is that better?
Yeah, that looks great.
Okay, terrific. Okay. Hi, everyone. Sorry about that. You can practice technical stuff all you want, and there's always something else. So I'm excited to be here, and I encourage everyone to ask questions, and it may be difficult for me to see them during the talk, so just go ahead and input them, and we'll go over questions later, and you can always reach me at a later time too if you realize later in the middle of the night, I wish I asked that question. Feel free to contact me. So, and I know that this series, you've had a fair number of talks about CADASIL, the clinical aspects. You've heard some fantastic research talks by CADASIL investigators studying both the sort of epidemiological aspect as well as the basic science. I'm going to focus on some of the genetic pieces that you might intersect with as patients. So like the genetic testing and how do we interpret genetic test results. So the outline is the following. I'll just a 1 or 2 slide introduction. I'll bring everyone back to high school for a genetics refresher. And then we'll just talk about the genetic variants in Notch3 and how we interpret them, and why we think certain variants are problematic and why certain variants are not. And then I'll just talk a little bit about my thoughts on genetic testing, because I am, at the end of the day, still a neurogeneticist, and those sort of decisions I think are very important. And I like to promote sort of a thorough understanding about those decisions. So CADASIL, just to bring everyone on the same page, as we know, it's very difficult to remember the full title, but it's cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. And those words are pretty much describing some of the changes we see on brain imaging. And it is a rare disease. It's caused by a single gene mutation in the gene called Notch3, and it causes a vascular type dementia, so cognitive change, as well as early onset strokes, seizures, some psychiatric stuff. And that's all related to damage in the parts of the brain that are fed by little vessels in the brain. So when I say variant, genetic variant, I'm also meaning mutation, just to let people know. So it's autosomal dominant, and recall that we have 2 copies of each gene. We get one from mom and one from dad. And so that means that if someone has CADASIL, like if this dad right here, I don't know if you can see my pointer. If this dad here has CADASIL, he has one Notch3 gene, CADASIL Notch3 gene, and one normal Notch3 gene, and his kids have a 50-50 chance of getting that Notch3. So kids with an autosomal dominant, at risk for autosomal dominant disease, have a 50-50 chance of inheriting the disease. So the Notch3 gene sits right here on chromosome 19. And again, there's 2 copies of the chromosome. And if we were to zoom in on that chromosome, we can see this example of nuclear DNA. So the DNA is all wrapped up, incredibly long, gets compacted into the cell, and sits in the nucleus. There's other types of DNA, but we're going to focus on this kind, the nuclear DNA. And then if we go and zoom in on the nuclear DNA, you see this, what you may have seen, this double helix structure. And so there is the outside rim of the double helix in blue, and then in between here, so you see these letters A, G, T, C. These are the nucleotides, and they form sort of the rungs of the ladder, and they fit perfectly together. So A and T match together, G and C match together, and each of these letters are stand for a certain title of a different type of nucleotide, and there are 4, A, C, G, and T. So when you get a Notch genetic test, this is an example of a real test result. You'll see that the report will say Notch 3, and that means that's the gene, and it gives a location on that chromosome, which does not mean anything to most of us. And then it'll have a description of the variant. And so it gives you 2 pieces of information generally. The first one here, which starts with a C, will be some number and then a letter, like in this case, G and A. And when they're talking about those letters, they're talking about these different nucleotides. They're talking about the switch from a G to an A. And you can imagine if the G here turns into an A, well, then this now turns into a T instead of a C. So there are consequences to these nucleotide changes. So we go out of the nucleus. Here's our nucleus. Here's our DNA. The DNA forms another intermediate step called RNA, and then the RNA is made into the proteins that we know. And so there are these molecules that bring the building blocks of proteins, which are called amino acids. And so here the amino acids are represented in spheres as these different colored spheres. And each of the amino acids, there are at least more or less around 20 of them. They each have different names. And so, for instance, here's an amino acid. It's a cysteine amino acid. We call it CYS, cis for short. Here's a different type of amino acid. And then the amino acids get strung together to form a protein. And you may have heard of the cysteine amino acid because it's very important in terms of Notch function, the structure, which we'll talk about. And it is by far and away the most commonly involved amino acid or change that causes CADASIL. And so when you're looking at a genetic test result, here's another piece of information you get. So this is P for protein. And here, this is saying instead of a cysteine, you have a tyrosine. So to take it all together, you have your DNA that has 2 different strands. It gets translated from RNA into these amino acids, and this amino acid string is what causes, creates your protein. In the case of a Notch 3 variant or a mutation, there is one change that instead of an A, it gets turned into a C, which then has consequences to the amino acid that it brings in, and then that changes the protein. So that's what this little orange here is. Now I mentioned the cysteines are important. So let me go back to that. What is the structure of Notch 3? So Notch 3 is part of a family of proteins that are very important actually for a whole slew of biological processes, development, immunity, vascular function, and these proteins sit on the outside of the cell or on the cell, basically, and then they interact with another protein, which I'll show you. And so this structure of this protein that sits on the cell is very important. And so here it is sitting on the cell. Now, if we go zoom in on this Notch 3, here it is lying on its side. Each of these little bars is called an EGF-like repeat domain. And I'm going to talk about those for the rest of the talk. So EGFR domain, there are 34 of them. They repeat throughout this region of Notch 3. Within each of these EGFR domains, so within each of these 34 rectangles, are some of these cysteine amino acids. There's a bunch of amino acids in here. The green ones are the only ones that are being called out because they're involving a cysteine. And normally there are 6 cysteines that sit at Notch 3. And what's important to know is that these cysteines, when they're actually in a protein structure, interact together and form what are called disulfide bonds. So they're physically interacting, and that interaction gives structure to that whole region. And so there are these 3 bonds that form, And in CADASIL, if there's disruption to the interaction of these cysteines, so if you have a mutation that makes you lose a cysteine or that makes you gain a cysteine, so for instance, here you might gain a cysteine, then you disrupt those sulfide bonds. And so why does that matter? Well, so here is what a normal region of these EGFRs would look like, where you have this disulfide bond. So this would be an example of here's what it looks like when you stretch it out. And then, but the disulfide bond gives it some structure. But when you have this mutation, you lose that normal 3D configuration of Notch 3. And is that important? That is. So now we're going to go zoom out. So here are the cysteines. Here's what happens if you break it up. You open up the EGFR domain. Now, these EGFs that are configured in a certain way are running all along the Notch3 protein. And so here's an example of that. And the specific structure or orientation of those different domains is what allows it to appropriately interact with the other protein it needs to interact with in order to function properly. So if you were to disrupt this structure, you disrupt the way the Notch protein works because it can't interact properly. And so as an example, so if this is no longer functioning, then you can no longer send signals from the outside of the cell to the inside of the cell. And so presumably this is why having a cysteine mutation is bad. And that is really what's been driving a lot of our understanding of Notch3 from a patient standpoint in terms of the genetic testing, that if it involves a cysteine and if it's in that EGF domain, we think it's probably likely to be causing a problem, assuming it fits with the clinical symptom. So like I said, so here is an example of a true genetic test that someone got that we were suspicious had CADASIL. And so you can see the variant it has the description of where it is. It shows you the variant. And then this lab called it likely pathogenic, which was consistent with what we thought. Now we had a different patient where again, we thought this person likely had CADASIL. And we did testing. Now here, the result is called uncertain significance. So why would these two different labs come up with different interpretations of a mutation that involves cysteine? And so this is why. And in fact, even in their description, they give you plenty of reasons why it should be called pathogenic. But I wanted to point out here that despite having similar approaches to why something may be biologically likely to be pathogenic, labs will have different thresholds for when they call it pathogenic or not. So if you see a lab report that has a stamp of pathogenic, uncertain significance, or whatever, just know that there's still some personal interpretation there. You should always talk to your doctor about what it really means, because it's not like everyone would say the same thing about the same variant. So how does a lab decide if a variant is pathogenic? I'm going to talk a little bit more broadly now, because again, this speaks to how a lab interprets these genetic tests, because, you know, and we now have genetic tests for a whole slew of diseases, and so I think it's probably relevant to understand how we think about them. So here's this sort of matrix. Each row is a kind of data that we use to evaluate a variant. So population data means how frequent is it in the population? If we're talking about a very rare disease like CADASIL, then the mutation that's causing that disease probably is also rare. In other words, if you find something that's in 20% of the population, it's probably not causing the disease that you know happens only 1 in 100,000. We use computational predictive data. So this is computer modeling to figure out, sort of like what we were talking about with the Notch structure, computer modeling to try and figure out what a certain change would do. Functional data. So that's really a pretty exciting field right now. Labs trying to make this higher throughput where we can take a variant or we can survey a whole bunch of variants in the dish and try to figure out which variant is most likely to cause problems. And so that's you're not just modeling it in the computer, you're trying to see how does it actually affect the way the cell functions. And then the caveat to that, of course, is that you have to be guessing the correct function of the cell. But so that's another way in which we look at mutations. Another piece is segregation data, and this is really important because we all have tons of variation between us. And so if I did my gene testing and I compared it to someone else in my lab, we would have plenty of differences, but would they make a difference? We don't necessarily know. But if you find a gene mutation that looks suspicious, you feel better that it's probably causing the disease if every person in that family who has the disease also has the gene mutation. And so that's what segregation means. And then there's some other pieces that have to do with the way the variant itself is being inherited, et cetera. So different ways that we assess the gene variant, and then the field has come together to decide if what constitutes strongly suggestive of it being pathogenic, being disease causing, or something that's strongly suggestive of it being benign, nothing to worry about. So they look at this matrix and they kind of map what they can map onto this matrix, and they make some decisions. And so using that matrix I just showed you, we then combine all the data. So you go row by row and say, well, what do we think this is? So this fits into this square of likely pathogenic, or this fits into this square of likely benign. And then it's a very complicated way, you add it all up, and then you can say, okay, well, based upon all this information I have, I think it's likely pathogenic, or I actually think it's a variant of unknown significance, which means that it has some pathogenic, some benign, or it has nothing. You just can't tell, and so you leave it as a variant of unknown significance. Or you might feel really confident, it's in 25% of the population. It's I already know that it's never been reported with the disease, and it doesn't do anything in the dish, so I think it's benign, and that's good enough to call it. So so that's what a lab does when it makes these decisions. Now, as the clinic, your clinicians are likely not probably just not going to take a look at the result itself. They'll want to do some of their own investigation. And there are now a number of fantastic databases that are available to clinicians and researchers that help us to aggregate all of this data, because obviously, we aren't going to have sort of a library of information about all those things. And so there is a database called ClinVar, where investigators and clinicians can enter what they know about a particular variant, and then what they know about the case, and then that gets uploaded into a database, and then other people can look and say, oh, actually, someone else has already reported this variant, and they saw that it was running with the family, and they think really that it's most likely pathogenic, so my isolated case probably is pathogenic too. So here's an example of a database that we use called Varsome, which is available to clinicians and investigators. And it actually kind of looks at all those pieces I was just telling you independently. So it'll look at the frequencies of the variant in exomes or genomes. So exomes are the exome is the 1% of that whole 3 billion pieces of DNA that actually gets turned into protein. The genome are all the billions of DNA or nucleotides. And so it looks at the frequency. Is it rare? Is it not rare? It looks at these in silico predictors. Like I was saying, when they do the computer modeling, they look at what has already been reported by other clinicians. And then, so here's an example of what the computer modeling does. So there's not even just one computer model, there's a whole bunch of different ones. And then they will each give their own score. And then there are ways that you can sort of aggregate these different scores and kind of get a general sense of it. More often than not, is it looking more pathogenic or is it looking more benign? And so here's an example. So this was a typical cysteine involving mutation, and it's thought to be likely pathogenic. And here's one that's been considered benign, so it's in green. And again, so it's there are computer predictions that suggest it's probably not going to cause much of a problem. And the in silico programs don't think it's going to be a problem, so it seems like it's likely benign. Now, here's an example of an uncertain significance, and this is always the one that's very difficult because so here the in silico predictions, so it's rare enough But if, whoops, oh, I didn't include that. But the in silico predictions were kind of either way. So some of them suggest that it could affect the structure of the protein and some of them didn't. And so the labs in those cases will just more or less say unknown. Now, in the clinic, what we do is we say, okay, this is a variant of unknown significance. We really can't give you any more information about that, but we will go back and look at that variant every couple of years. And sometimes the lab will reclassify a variant as actually being pathogenic or not pathogenic. More often than not, the variants go from variant of unknown significance to benign, but sometimes they can go the other way. So when we think about how to classify a variant, there's two big things that are sort of embedded within that matrix, which is what does the variant do to the protein structure itself? And so you can have computer models that tell you. And then where is the variant in the protein, which again is going to speak to how does it affect the function. And more than 98% of the variants that cause CADASIL involve a cysteine amino acid. But that means at least 2% are not, and I'm sure it's more than that. We're still learning more about them. And then there are at least more than 200 variants, and they're all across that domain that I was talking about. So here's an example of a person with sort of typical CADASIL. And you all may have seen these MRIs. This is sort of a classical MRI where these are the temporal lobes. So where there's white signal, that's the abnormal signal. That's where there's been tissue damage. And so this anterior temporal lobe, which means the front of the temporal lobe, that whiteness is typical of CADASIL. And then right here, there's this called the external capsule. This is where fibers are coming in from the neurons up in the cortex, and they're coming through. And then here again is where some of those fibers, the fibers that are being affected are now white because there's damage. Now, here is the MRI of someone who looked like he had CADASIL in terms of clinically he had early onset stroke, et cetera. But his variant did not involve a cysteine. It was this R75P. And while he didn't have really a dramatic presentation, or his MRI wasn't dramatic, it still had some of the features of the brain MRI that looked suspicious. And it certainly is, I think this is an important point, certainly is much more affected than you would expect for a 60-year-old who just had, no significant medical history. So it turns out that that these cysteine sparing variants actually have many features of CADASIL. So here this is from a paper that actually had a much longer list, but this R75P that I just showed you is probably one of the more studied. And you can see, so on each of these rows or each of these columns are different features that we typically associate with CADASIL. And you can see that these individuals who all had mutations that did not involve a cysteine, they generally still had stroke. They had involvement, the pseudobulbar involvement that's associated with stroke, that type of stroke. They may have had cognitive changes. And importantly, a number of these people that had the cysteine sparing also had the GOMs. So the GOMs are the granular osmophilic abnormalities that you see on a biopsy. So this is the vessel abnormality that can be taken from a skin biopsy or anywhere really. And that used to be sort of the way that we would diagnose CADASIL. It's not 100% sensitive, but it is, but it can make the diagnosis. And so having this abnormality on pathology is another supportive feature that this is likely pathogenic. So that's, so that makes sense. Some of that stuff is consistent with having CADASIL, but are there other ways to do this? And you may have heard about this. So a CADASIL researcher, Dr. Michael Wang at University of Michigan, has really been focusing a lot of work on trying to understand Notch3. How does Notch3 variants cause CADASIL, affect the vessels, really getting to the core of the basic biology of the disease. So what I'm going to show you is something that is called a Western blot analysis. And what that is it's a way to separate proteins. So you can, take a piece of skin or you can take a cell, and let's say you want to know what are all the different proteins, and does one cell have more of this protein or the other protein? What you can do is take that mess of proteins and put it on top of a gel and run a charge through the gel. And then based upon the structure and the size of the protein, it will move down the gel differently. So here, what you see here these are all different proteins, these little black bars. And the ones that are lower are the smaller, faster ones, and the ones that are really big were slower and bigger. So what Dr. Wang did was systematically look at all the different residues, all the different amino acids in that EGF-like domain. And so what he's done is create different versions of the Notch3 protein that have either typical mutations for CADASIL, so like a cysteine involving here, or this R75P that I was just talking about, or ones that are that were not likely to be pathogenic. So ones that we weren't suspicious of, like this 113 and R75. So what he did is he created all these different types. So the protein he made doesn't have all of them at once. These are just different versions of the protein. He took them and then he ran them on this gel and he looked to see how they move through the gel. So the WT is the wild type, and that's just the normal notch. And if you take that notch protein, you put it through the gel, this is where it lands. Now, if you take a CADASIL version of notch where you make this change, that structure changes the way it moves through the gel. And so you see that with the typical CADASIL mutation number 1 here, I don't know if you can tell me, I see my pointer, number 1, this is where number 1 is moving, and you can see that he was stuck way up here. And the different variants all move differently across down through the gel. And what you can see is that some of the variants, like the ones over here on the right, are the ones that we weren't so suspicious of because they didn't involve a cysteine. They probably didn't have a lot of clinical data to support that they were pathogenic. And you can see that when he created notch proteins that had these variants that we were not so suspicious of, 8, 9, and 10, which are 8, 9, and 10 here, they moved through the gel just like the regular wild type did. So this was a way that Dr. Wang could evaluate mutation by mutation, how it's affecting the Notch3 protein itself. So not just in the computer, but actually physically on this gel. Another way that people have been trying to understand how the Notch protein changes, or how the mutations change the protein, and also how do you distinguish between a mutation, a pathogenic mutation or a benign mutation. So this is another more of a biochemical assay. And I think what's important to see here is the higher the bar, the more aggregation there is. They start with a single molecule and they look at how it aggregates. And the Notch proteins that have the typical cysteine mutations aggregate more. And so these little colors are how long you let it sit. So if you let a protein sit, it'll with time it will start to get clumped together, and the CADASIL ones tend to clump together more. The variants that we know again are likely not causing any problem. They don't clump much. And then some of these other ones again that don't involve a cysteine but are suspected of being mutations do have that aggregation. So that's a cellular way that's a biochemical way to look at these mutations. So how else? I've been sort of talking about these EGF domains a lot. What does it mean to have a mutation in a certain place? And it looks like, let's see, maybe I can't actually see the Q&A. So whoever has a question, hold on to it. There we go. Okay. So the interesting thing about, or one of the many interesting things about CADASIL is that we have learned a lot about the disease thanks to folks like you who have been willing to donate their time to research studies or have been coming to the clinic and seeing us regularly so that we can learn more about the disease. And so some of the features or some of the characteristics we give to CADASIL is biased a little bit in a way, in that it's really based upon the people that come to us in the clinic rather than just sort of a broad swath of people out in the out in the world. So there's something to be learned by looking beyond the clinic. And so this was a study done in 2016 that was looking at different types of mutations that could be identified in databases that were not involving CADASIL patients. And so, as you can imagine, over the years, there have been a number of studies at looking at diabetes or looking at weight or just, I'll show you another one, just studies that are looking longitudinally at general health. And there's been a lot of genetic data. And so there's a way now that we can look at that genetic data to help us learn more about the diseases that we're interested in. And what we knew already is that there's hotspots or regions where most of the mutations are most likely to happen within that Notch3 gene that are causing CADASIL. So more of the CADASIL related mutations happen really early in this EGF-like domain 1 through 6. Now, when this group went on to look at other CADASIL databases, they found, or worldwide databases, they also found that in addition we knew this already, sorry, that there are different hotspots, but the people that had mutations in this area here, in this part of the gene that we're going to call EGF 7 through 34, the people that have mutations here tended to have a different type of disease than the people that had mutations here. And so what they did is they looked at these folks that have CADASIL, and so and they looked at the percent of them that were free of stroke. And so the people that have typical CADASIL were in blue, and the people that had the mutations later on down the protein are in red. And you can see that there's a shift that, say, the number of people that are 50%, the 50% of people that are free of stroke is shifted, that by age 50 or so, 50% of people have had stroke with the typical mutations, whereas it's not until the 70s that people with the later mutations, that 50% of them are free of stroke. Now, so that's just like on a clinical description, but there are other biomarkers that were also supportive of that. And so we were talking about the MRI, and these are, again, the sort of typical CADASIL changes. This is an image of someone who had one of these later mutations. And you can see that, again, he has some of these features that are typical of CADASIL, but not necessarily as much. And so on this graph, the study had quantified the amount of this type of damage and showed that in the individuals with typical mutations, they had a higher amount of this brain MRI abnormality compared to those that didn't. So is it possible that there are people that are so mild out there that they don't even know they have CADASIL? So that's one question. So what folks have done is then go to look at large international databases and say, okay, well, where do we find Notch3 variants even in the general databases? And they did. So in CADASIL, like I said, there's mutations that happen in these regions. When this study looked at a large database, they found that even in individuals who didn't have a clinical diagnosis of CADASIL, there were mutations that involve a cysteine that were also in the Notch protein, but just had never come to clinical attention. So another way to study this is to use these, like I said, these databases where people get phenotyped, and so they get characterized really well. They volunteer to get brain MRIs regularly. They get clinical exams, and they donate blood to have their genetics done. And so there are different layers of genetics that have been done on more than 500,000 people out of this place called the UK Biobank. And so looking at those 500,000 genomes, Dr. Rutten and her group have learned even more about what it means to have these sort of later mutations. And what they found is that, again, if you take a look at people that have CADASIL, the ones that have the 7 through 34, so the later ones, again, had that shifted later onset of stroke. But then there are people in the UK Biobank that have those same mutations and they actually don't have a lot of stroke. They have some, but not necessarily, but not like you'd see in the group of people that come to us in the clinic. And they actually were able to quantify this. So what this is trying to show you is that someone that was found to have one of those more benign mutations, this is what his MRI looked like, and it was pretty, pretty unremarkable, and which led the investigators to say, that there are people out there that having a mutation in Notch3 that you suspect is causing disease may not always show itself. And so these are scores, again, looking at the brain MRI, and the higher the score, the more damage. And so you can see that these folks in the UK Biobank that had the CADASIL type disease mutations, they did have changes to their brain MRI. So they weren't completely normal, but they just weren't clinically they weren't clinically coming to attention. So, okay, well, I just want to mention for testing that there are different things to think about when you do testing. There's considerations for yourself about diagnosis, about treatment. Do you take aspirin? Do you want to let your emergency room know that you have CADASIL in case something happens? You can wonder more about prognosis, but what I want to explain too is that some of the data that we knew from decades ago about life expectancy, et cetera, are probably not as accurate as we think, and it's probably much more variable. And I think it's important to know that, especially if you go and Google, sort of older studies. And then for the family, you get tested. It has implications to your parents if your parents didn't know if they had CADASIL, so it's always something to think about. And then genetic testing can be helpful for family planning. You are protected from discrimination at the workplace and for getting health insurance. So even if you don't have symptoms, but you have a gene test positive, you cannot be rejected for health insurance, or you can't be discriminated at work. And that is actually not the case for all types of insurance. And genetic counseling is really important. These folks go and they get a master's degree in genetic counseling, so they're very knowledgeable in the science as well as how to think about, do I want testing or not want testing? They can help people think about, when's the right time to test, et cetera. And sometimes, you don't necessarily want to test at that moment, but you just want to know more about it. And that's what genetic counselors are great for doing also. So just to remind people, so you cannot be discriminated at the workplace or with health insurance, but you can be discriminated against, or you can be denied long-term disability and life insurance. And so if you're thinking that you might want to do that, you might want to get that in place before you do genetic testing. And so I'm going to stop here and just say thank you to all of you and for being involved and trying to help us cure this disease. And if you have questions, I'm sure there's contacts from the CADASIL Consortium that we can help triage. You can also try and help me. So call me. So Iman, should I turn off my screen here?
Yeah, whichever way is easier for you to answer the questions. I think we've gotten a couple so far in the Q&A box.
Okay. Go to the Q&A box. Where am I? Uh-oh. Okay. Shoot. Oops. All right, here we go. Okay. Oh no.
I can read them for you.
Can
Do
you?
you want me to just to read it?
Yes,
Yeah.
please.
So the first one is, are there other mutations caused by Notch3 mutations? And that's all they said, but I don't know, maybe you want to talk about non-cysteine altering mutations or anything about that. But
yes.
The questions are there other mutations caused by Notch3 mutations?
Are there other, and the person doesn't mean are there other diseases, right?
No, I think what they mean are there other maybe other types of Notch3 mutations.
Yeah.
So the way I interpret the question doesn't quite make medical sense, and I don't blame the attendee.
Other diseases, okay. I think that
but maybe we can ask. Yeah, well, so the question could be one of several. It could be Are there other diseases caused by Notch3 mutations?
Yeah.
Are there other types of mutations other than cysteine altering that might cause CADASIL?
Yeah. Yeah. So those are and different diseases that can. So there are different diseases that that the Notch genes can cause, and so there are there's a collagen vascular disease that can be caused by different Notch protein, but Notch3 protein, the Notch3 gene itself, is typically just CADASIL. And but yeah, but the other notches are also very important and cause different vascular diseases.
And then oh, sorry. Can you see the questions yet? Or do you want me to keep
I
reading?
can see, yeah, I see the other. I don't know why it's so difficult for me.
I'll
but
read it.
like
Do
I see
family
the other
members who have
part.
CADASIL always have the same mutation?
Yes. So okay. Let me go back to one more thing though, which was the different genes. I should mention that there are different genes. So we talked about the mutations that were in cysteines, but there are other gene mutations that can cause something that looks just like CADASIL. They can be autosomal recessive, where you have to have two mutations that cause that. You get one mutation from dad, one mutation from mom, and then you can get the disease. And then there are a few other rare diseases that can look just like CADASIL. Does everyone in the same family have the same mutation? Yes, they do. So generally that the mutations happened in one person and one relative way, way long ago, and then each person inherited that block of DNA. So, I may have inherited it from my mom, and then my cousin may also have it, but he has the same version because we both got it from our grandmother.
And then how do I go about genetic counseling? I have CADASIL and would like to know about what my specific mutation means. I would like to know if my mutation is in EGFR domain 1 to 6 or 7 to 34.
Yeah. So there are different ways to do that. If you have a neurologist or someone that you see for CADASIL, that person, him or herself, can talk about it with you if you have the gene report. Now you need the gene report, the test report, to know exactly where the mutation is. If you're looking for a genetic counselor, there are ways to do that. So if you're near a university setting, or and I guess not necessarily even university, but a larger academic or larger medical center, they will have genetic counselors in a genetics clinic. So you can always go to medical genetics clinic. If you're in an academic center, some of the stroke folks may also have genetic counseling or be able to talk to you about it. And then this other option, which is, I can't really necessarily say much about, but I know is available, is that there are direct genetic counseling services that are available, and I'm happy to work with people to help find something if you're not near a center. And then Iman is reminding us that the CADASIL Consortium itself provides genetic testing and counseling to people who have not been tested. So we are very committed to helping you go through that process and informing you, keeping you as informed as you can. But yeah, if you already have the test, we can talk about it. And
can I add something to that?
Yeah, please.
Yeah, so there's Dr. Geschwin here. Most genetic counselors who don't specialize or know about CADASIL will not know the minutia about the different EGFR domain, epidermal growth factor domains that Dr. Jayadev talked about. So they won't really be able to easily tell you the difference. First of all, they may not. They can figure out if you tell them in advance you want to know it, but on the spot they're not going to be able to quickly tell you what domain, what EGFR domain your mutation is in, or if it's even in an EGFR domain. If you let them know in advance before you meet with them that that's the type of question, then they can look it up. But most won't even know about many of the papers that Dr. Jayadev stated, because they're just not going to be as updated as a person who studies CADASIL and does research in CADASIL is. But the more you prepare them, the more you tell them about the questions that you have for them, the better prepared they'll be to answer your question.
Yeah, really great point.
So I see one of the questions is, if I look at my findings, how can I determine my mutation number? Do you want to talk about that?
Yeah, if you mean, how do I know what my actual mutation is? It would be on a lab report, and we can talk about, so it should be where it says variant. So it'll say results, and each lab is gonna be different, but it'll say variant, and it'll have a C dot blah blah blah, and then a P dot blah blah blah, and that tells you what your mutation is. And then if you want to know where it is, then that becomes much more complicated, like Dr. Geschwin was saying. And so I think the important part, I think that, more important than I think the location, because I think what I probably needed to convey is that those later mutations can be found in the general public, which means that not everyone has, strokes at age 50 if they have these mutations. But a lot of the people that we have in our clinic with typical CADASIL have mutations in those later regions. So it's not like everyone who has mutations in the later region has mild disease. It's just that they also exist in the general public. So in terms of knowing exactly where it is, I think you're better off talking to someone who can look up in the databases for you, or look up on the gene maps exactly where it is. But I don't think it necessarily helps you on a personal level, medical level, prognosis level. It's more, all these things are sort of useful for researchers to understand the disease. When we're looking at thousands of people, we can kind of come to some sort of conclusion, but that information is not useful to us on a one-on-one basis, on a person-by-person basis, because there's such variability that, I might find an association if I have 50,000 people, but there's such variability that I needed 50,000 people in order to find that difference, which means that if I'm trying to understand that one point in one human being, it's almost impossible, because I don't know where that person would be in that big variance.
Another question that came up, somebody said, are there any theories as to why some non-cysteine altering mutations in EGFR 7234 cause clinically significant disease in some people, but not others? And I think you might have already partially answered that, or completely. Or do you want to?
Yeah, and I think there's a lot of different ways to think about that. So one is, why is it that some mutations in that region cause problems and some doesn't? And I would say, first of all, it could very well be, a lot of it is this, how does it affect the structure of the protein and how doesn't it? So one mutation, if it's pretty not a significant change in terms of the charge or the size, it may not affect the protein. Whereas even in the same position, if it was a different type of amino acid, it might significantly change the structure of the protein. And so it may change whatever it is that's happening that causes disease. And what is happening, I think, and we're still working on it, but let's just say it's because there's too much of this extracellular piece of protein, or that it aggregates more. So some mutations might cause it to aggregate more and some may not. Now, but let's say you're asking, well, I have this mutation and my next door neighbor has the same mutation, but we have very different presentations. Why is that? So another an important piece of how a gene mutation turns into a clinical presentation is has to do with also all the other 19,999 other genes that we have in our body that are, more specific to us. And so we know that there are genetic modifiers of disease that are, sort of independent of CADASIL, but influence the way our blood vessels work, or influence the way that we interact with our environment. And so those genetic modifiers can also explain why there is such variability in the way the disease presents, even in the setting of the same gene mutation. That's a good question.
And somebody asked about, can you donate plasma if you have CADASIL? And most research studies in CADASIL will accept plasma. So our study that we're doing, the CADASIL Consortium, the one that Dr. Jayadev, I, and others are part of. We collect blood, and then sometimes blood can be separated into different components, but we collect blood, we collect, including plasma and serum. And then other studies also are very interested in different blood, in collecting blood, including plasma. So, I think Aman just said, can you donate plasma if you have CADASIL? And so I think what they mean is, can you be a blood donor,
Right.
maybe? So if that's the question, yeah, we don't think that CADASIL is transmissible. There are some neurodegenerative diseases for which you cannot donate blood products, such as prion disease, which is one of the diseases I study. But for CADASIL, there shouldn't be any issues with donating blood. You should be able to donate blood. But if you do want to donate blood for research, there are studies. We have one, NIH has one, the Chan Zuckerberg with Dr. Elahi and Mashia have one. So there are lots of opportunities to donate.
Yeah, and those that plasma, the donation of the plasma to research is actually very helpful to understand some of the questions that we've been asking. You know, why do some people get disease, some people don't? Why is this particular mutation a problem? And then so we can learn what changes in the blood before anyone has any clinical problems. And then importantly, once we get a drug, we can actually follow how people are responding to a drug just by looking at their plasma. I mean, that's the hope.
And I see another question that I think you could answer, Dr. Jayadev, which is, someone said that they tested negative for CADASIL, but how confident can they be about that with so many others in their family that have tested positive?
Great question. So, you know, in general, I think the technical aspects of the labs are really strong. And if I were to give a negative test result from a lab that does this all the time, I would do it confidently and say, you don't have it. One way that sometimes some clinics will do it is that they will try to make sure they send the sample to the same place, the same lab where other family members have been tested, just to make sure that the methodology they did to identify other family members was used on this person as well. But if you test the negative, I'd feel confident, pretty confident that it would not pretty. I would feel confident that it was actually negative if it was done in a regular, bona fide clinical lab.
Great. And then another question is, I guess geared specifically to your lab, Dr. Jayadev, but does your research include the HTRA1 diagnosis, and how similar is it to CADASIL?
Yes, that's a great question. So my lab, I don't actually study that protein, although interestingly, it had come up for something I was doing like 18, 15 years ago. But anyway, but it is, so I don't study it on a research basis, but it is on the tests that we do for people. So if someone comes in and I'm wondering if they have CADASIL, but they don't have a family history, that is a gene that we think about and we can test for it. Yeah. Is that?
Yeah, I think that's great. Another question came up, which I think you can kind of gear on the variability of CADASIL presentation, I guess, in families. It was asked that, what does it mean when neither parent was known to have CADASIL, but one child of 5 does have CADASIL?
Yeah. So there are number, so that would mean, so there's a couple possibilities. One possibility is that Joe Schmo has CADASIL, or Joe has CADASIL, he has a G mutation, and his parents are perfectly healthy, and they don't seem to have CADASIL. And in that case, it's possible that one of them still has a mutation but doesn't have disease. But I imagine what you may be asking about too is that Joe has the mutation, but his parents have been tested and they don't have the mutation. How is that possible? So there are different ways that this could happen. One is that it's de novo, which means the mutation just happened in Joe himself, and it was the first place that it started, and it means that if it happened in him, that means that none of his siblings would have had, are unlikely to have the same problem, but his kids would still have a 50-50 chance. It's also possible, this is getting very much in the weeds, is that it's possible that some of the cells in his parents had the mutation and some didn't, and the ones that formed the sperm or the egg were the ones that had the mutation. And so mom or dad may be mosaic, which means that some of their cells have it, some don't, and he inherited some of the DNA that had the mutation. But so there are different reasons why that could happen.
Great. And then I think we have time for just one more question, which is, can active immunization lead to reduced levels of Notch3 extracellular domain in the blood via immunotherapeutic approaches?
Yeah, and Dr. Geschwin may have a better sense of this. I know that there are people that are interested in this concept, that there is the mutation in Notch3. Now we know that Notch3, as part of its activity, gets cleaved at the cell surface, and then the extracellular part, so the part that's no longer connected, kind of goes away. But in CADASIL, we see it aggregating, and we see it in the blood vessel. So is it possible to use an antibody and bind that aggregate and then have your immune system clear it? And I don't know where that is conceptually or in a trial form. Michael, is that?
Yeah, I'm not aware of, it's certainly a mechanism which some laboratories are looking at for trying to get rid of the disease. One of the problems is that it might be too little too late. We have a similar problem in Alzheimer's disease, where we get a lot of clumping or glumping of amyloid protein. And there have been dozens and dozens of trials to remove amyloid. And right now, only 2 have showed any promise. And one has led to an approved drug, but it's a pretty weak effect. So we're not so sure how, even if there is an antibody that can remove the Notch3, will that be sufficient? But it is one of the areas in which investigators are looking at, trying to think about at what part along the pathway of the production of this protein do we want to attack the disease.
Yeah, great question.
Great. Yeah, really good questions.
Yeah, these are all been really terrific, guys. Thank you.
Well, it was a really great talk. There are a lot of comments in the Q&A that are just talked about how clear and helpful the talk was. So,
Oh, great.
Dr. Jayadev, thank you so much.
I'm glad to hear it.
Yes.
All right.
Great. Thank you everyone for joining. And if you have any follow-up questions for myself or Dr. Jayadev, feel free to email me. And thank you all for joining this evening, and I hope everyone has a wonderful night. Thank you. And thank you, Dr. Jayadev, for the beautiful presentation.
Thanks. Great. Bye-bye.